Hello everyone. Today I will present you our recent study, few-shot protein function prediction with multimodal representation learning, and our tool, holistic protein representation, HOPER. As you all know, protein function prediction is still an open and challenging problem. The number of new amino acid sequences grows rapidly. However, number of functional annotations for these sequences couldn't match. Hence, a large number of functional properties of proteins are still unknown. This creates a gap between protein sequencing and annotation. In the CAFA challenges, protein function prediction methods evaluated regularly, and the results show that protein function prediction is still an open problem. This problem is more significant for less studied functions and proteins. We address this low data scenario in our study. In the literature, there are attempts to solve this problem. However, there are only a few methods exist. Most of these methods solely use protein sequence as input, however, does not take advantage of multimodal learning and other modalities. Methods such as ProTranslator takes advantage of these modalities such as sequence, text, and PPI, but does not complete an integrative modeling approach such as multimodal learning. Currently, development of holistic approaches for protein function prediction is a need. Current solutions require training and fine-tuning new models from scratch for each task, which is costly in terms of time and money. So we can say that these solutions are not problem-specific on low data. Also, validation of these methods on real-world clinical cases is another need here. Here our claim is, under low data scenario, learned protein representations can be a solution, especially with multimodal learning. For this goal, we first developed PROP, a reusable benchmarking framework for evaluating different protein function prediction methods, which is a comprehensive benchmark tools and provides a reusable, unbiased framework for evaluating and comparing various protein function prediction methods across different dimensions. With PROP, we aim at systematic performance evolution, which guides researchers in identifying areas for improvement in protein function prediction methods. And with HOPER, holistic protein representation, we developed a novel multimodal sequence-based protein function prediction model. This multimodal model is a significant advancement in protein function prediction with potential to improve understanding of protein functions. This tool combines protein sequence data, protein literature text, and protein-protein interaction data. We utilize machine learning and language processing techniques to learn complex relationships between different modalities. Hence, we aim to improve the accuracy and efficiency of protein function prediction by addressing low data problem. Here, you see how protein representation learning methods work generally. They start with different data sources, such as gene sequences, protein-protein interactions, gene expression profiles, and biological literature data can be utilized for these models. And both unsupervised and supervised learning methods are used to create protein representations, which are basically vectors that represent proteins. Once these protein representation methods developed, protein representation vectors can be calculated for each protein and used for different tasks, such as protein classification, protein activity prediction, and protein design and engineering. This reusability is one of the important advantages of protein representation learning. And in the prop, we achieve two goals. The first one is an extensive review of the literature and benchmarking on four different tasks, which we will see shortly. We will evaluate human protein representation vectors for these tasks. Methods in current benchmark study are both classical and representation learning methods for proteins. Under classical representation methods, we evaluate homology-based methods such as classical baselines, as like blast hammer, and rule-based methods such as unirule to go, composition-based methods such as amino acid composition AAC, and sequence-based representation learning methods, but also other alternatives such as gene expression or gene co-expression or mutation-based representation learning methods on semantic similarity inference, ontology-based protein function prediction, drug target protein family classification, and protein-protein affinity estimation. Here you see two example results from our evaluations. Here you see semantic similarity estimation of different methods by using two different data sets, which include semantic similarity information but different sparsities, and also protein binding affinity prediction by using Scampi datasets. And here you can see our methods, representation learning methods evaluated, can surpass state of the art and baselines. When we inspect the results of probe, we see that protein representation learning models can be reused for a variety of tasks. This creates advantage of modest resource use requirements and high representation performance. We also observed superior performance of learning-based methods, which learned representations outperform classical models in functional analysis of proteins. Large-scale models perform particularly well. Classical methods such as Hammer showed a competitive performance with deep learning-based methods in molecular function category of protein function prediction. And we can say that incorporating homology information into training may improve predictive performances of representation learning methods. We also can say that, unsurprisingly, technical model design and trained data sources are two most critical factors here. But also, we should warn you for potential data leaks, such as one of the methods, Gene2Ec, utilized a hyperparameter optimization task which aims to maximize the clustering of genes with molecular signature database (MSigDB). Or another method, BERT-PFAM, employing sequence fragments that correspond to protein families in the supervised training process. Hence, this might cause an additional performance for protein family prediction. Okay, so what we did afterwards is development of HOPER model. In the holistic protein representation model, we utilize multimodal learning for protein function prediction, and we emphasize on low data scenarios. We started with multiple modalities as protein sequences, protein-protein interactions, and biological literature. For each of these models type, we evaluate different types of representations using our prop model on protein function prediction. Afterwards, we inspect these benchmark results and choose the best alternative for each modality. By using the best protein representation alternative for each modality, we calculate representation vectors. By using these representation vectors, we assess different alternatives, such as concatenation of these representation vectors or training of alternative multimodal deep learning models, and assess the results on two different cases. The first one is a more hypothetical one, which is ontology-based function prediction, and a real-world case, immunoscape protein prediction. So, in sequence-based representation site, we evaluate 23 protein representations using prop benchmark. We apply a linear classifier here to focus on representation performance. For results in predicting gene ontology terms, we can say that uh, learned representations mostly outperform classical methods, and but performance degrees with fewer annotated proteins, which is loaded as scenario, is widely observed for all types of methods, especially for specific or informative gene ontology terms. And we can say that top performing methods showed a consistent performance across all goal groups, which is the top performing method is ProT5XL on protein function prediction case, is a T5 based encoder decoder based large language model. For protein-protein interaction-based representations, we evaluate two widely used graph representation methods, HOPE and Node2Vec. Here, Node2Vec outperforms HOPE on protein function prediction problem, but both models are most successful in predicting molecular function category. Overall performance of protein-protein interaction-based models are lower than sequence and text-based models. One important issue here might be the mismatch between HOPE's main objectives and the use case, because HOPE mostly focused on direct graphs. However, here our data source, in fact, includes undirected graphs. Moreover, here we can say that our performance or model's performance are mostly sensitive to hyperparameters. We observed this in hyperparameter optimization, but we couldn't observe any specific correlations here. The next data type we utilize is text. Here we use UniProt flat files, which includes valuable created information about proteins, and PubMed abstracts, which are related with each protein we evaluated. Here, a surprising result is performance of TF-IDF, and we applied PCA over this TF-IDF vectors, which are very simple methods both, but in a combined way they outperform more complex models such as BioBoard, BioCentric, and BioBoard, which is a large language model. And a combination of UniProt and PubMed data as input yielded the best performance for TF-IDF. And more interestingly, BioBoard, the most sophisticated model, performed the worst. Here we can say that natural language processing approach successful or short sentences, but may not be suitable for longer sequences. This observation has been done uh, in different studies. So there are more specialized models for longer sequences like long former and reformer. However, these models have not applied or pre-trained on biomedical text until now. And similar to other modalities, all models underperformed in the low data category with no clear pattern for other categories. On the multimodal representation development side, we start with a baseline fuse trap, which is basically consists from concatenation of representation vectors for different modalities. Here, these are protein-protein interaction, PPI, text, and sequence, as we discussed. This second method is an simple autoencoder, which reconstructs input representation as a whole. And here, the input representation is the fuse trap, which is the concatenation of different modalities, as we mentioned. Uh, and for this model, we used bottleneck layer as protein representation, which we did the same thing for all other models. The second model we developed is multimodal autoencoder. And here the difference is, as you can see, this model reconstructs each representation separately. And we hope that the common information summarized in the bottleneck layer, which is used as the protein representation vector, as we mentioned. In transfer AE, transfer autoencoder, we aim to develop a sequence-based model, since especially for understudied proteins, text and PPI data don't exist. So we start with transferring weights of the multimodal autoencoder to a new model and train this model to reconstruct each of these modalities, PPI, text, and sequence, but this model only uses sequence as input. So what we see when we inspect the results of multimodal representations? First of all, we can say that unfortunately none of the multimodal models did outperform text in general and for low data. In general, simple autoencoders worked best for molecular function. TF-IDF was the best for biological process and cellular component. Key goals were developing a multimodal model for low sample functions, which means 5 to 30 protein samples for each annotation, and assigning functions to other studied proteins with only sequence data. Here, fortunately, we can say that these goals were met with transfer autoencoder model. As you can see, transfer autoencoder surpasses best sequence-based model for T5XL in all three categories of Go: molecular function, biological process, and cellular component. Here we can say that multimodal model has limitations, first of all. These models were not able to fully leverage combined power of different data types. Synergistic effects were not achieved. But we think that since we used relatively simplistic models here, there is still room for improvement. But on the other side, transfer autoencoders' potential can be seen here. Transfer autoencoders' improved performance in low data settings indicates its potential for addressing the challenges for protein function prediction with limited data. This approach could be beneficial for studying less well-characterized proteins. In the future, exploration of more advanced methods to integrate multimodal data to achieve better performance in protein function prediction will be important. Moreover, investigation of potential data leak from text to gene ontology terms and its effect on evaluation of multimodal models is critical. We know that such a claim is not easy to explore, but it can be done with appropriate tools. Finally, development of pre-trained long sequence models specialized in biomedical text to improve text-based representations for protein function prediction is another open problem. On the case study side, we aim immunoescape protein function prediction for lung adenocarcinoma. Here we start with TCGA dataset of lung adenocarcinoma RNA-seq data to identify different differentially expressed genes for lung adenocarcinoma and find approximately 1,400 differentially expressed genes. And we create multimodal protein representation vectors by using protein sequence, PPI, and text data using our multimodal autoencoder model. On the classification side, we evaluate different alternative classifiers such as FCNN, Fully Connected Neural Network, SVM, KNN, and random forest. For the training set, we gather 26 positive samples from literature and develop a negative sample dataset by using gene ontology based similarities and semantic similarity calculations. And here we used 124 negative samples with text, sequence, and PPI data. We also accomplish hyperparameter optimization using five-fold cross-validation for this task. At the end, we predicted 139 new immunoescape protein candidates and evaluate each of them specifically using literature search and functional annotations. Of course, we couldn't talk about each here, but for example, one of our predictions, MMP9, can stimulate second release and T regulatory cell production, which increase MDSC numbers. Hence, it might cause immune escape for tumors. One other prediction is rank L. Rank L here is a cytokine of the TNF-alpha cytokine family, secreted by tumor tissue and might create an immunosuppressive environment through activation of some intermediates, uh, which causes regulation of TGF-beta, and TGF-beta binds to T-cell receptors, and activation of FOXP3 is triggered. Finally, development of Treg cells occurs, which finally causes immunosuppressive microenvironments. We also evaluate some of our negative examples, since immune escape involves numerous proteins, and we observed that our negative examples or our model's negative predictions are solid. So, what we learned from the case study? First of all, effective representation models can be useful on real cases, such as immune escape protein prediction. Also, importance of developing better models is obvious here. Multimodal data could enhance accuracy with combination of sequence, PPI, and text data, which ends up with increased predictive power for immune escape proteins. We can also set that all our classifiers performed well, but these comparable results highlight the importance of representation quality. Also, we can say that there's an untapped potential for immune escape protein discovery because there's limited knowledge in current literature, and need for further experimental validation and research is important. But we can also say that more improved models can also help to identify novel therapeutic targets, not only in cancer, but also for other diseases. As a conclusion here, we can say that multimodal protein representations can address limited training data challenge. Benchmarking tool for low data protein function prediction was important, and we showed this importance in a case study for immunoescape proteins in lung adenocarcinoma, which we predict new proteins here. On text-based representations, we can say that these representations most outperform multimodal model ones. However, unexpected results might be because of potential indirect data leak here. Also, for large language models, alternative pooling methods are needed uh, for extracting knowledge from large texts, such as biomedical ones, and specific pre-training is needed here. On low data protein function prediction results and transfer learning side, we can say that multimodal methods are promising for low data protein function prediction. Uh, and we also think that Sequence-fed multimodal approach for low-data protein function prediction is still an open problem, but has potential to solve with these types of multimodal approaches. And for immunoscape protein function prediction in adenocarcinoma, uh, it's obvious that our models could reduce time for manual selection, and advanced methods using autoencoders and transfer learning has potential to change this field. In the future, exploration of advanced models such as GNN, variational autoencoder, or GANs for improved performance in multimodal learning is important. Also, graph neural networks for protein-protein interaction representation learning is another potential enhancement field. Moreover, as we mentioned, specific methods on biological text is needed, and development of modern fusion strategies and multitask multimodal learning framework for complex model interactions is a need and has potential. Finally, Prop is a tool which provides an extensive benchmarking platform on many different tasks, and we able to create Hopper by the Prop's detailed performance on low data protein function prediction evaluation. And here we Hopper facilitates deeper understanding of protein functions, particularly for less studied proteins. This also could be seen in our case study, which has potential to accelerate the discovery of novel targets on immune escape proteins. You can find our tools in GitHub repos in a reusable way. And I would like to thank you to all our team and National Science Agency of Turkey, TÜBİTAK, to support our work. Thank you very much for listening.